of homework number 1. So you're going to go to this site, you're going to select a conserved orthologous gene, something that's found in lots of different creatures. You're going to make a document, and like Pages is fine, or Word is fine, or anything that can save as .doc or .docx. You may want to check first. You're going to write down the name of your orthologous gene, sentence about the function, you can just Google that, and accession numbers for 10 homologs. I'll show you how. Run them through Protein Blast, screenshot your BLAST result and your BLAST stats page, and I want your BLAST alignment as a flat query anchored alignment, please. Then briefly analyze it, save it as a document, and turn it in via Canvas. Easy? I don't know. It's got a lot of steps, but it does fit in a fairly small space, so let's unpack it. All right, let's go to this site to find some conserved orthologous genes, and that site would be, here we go. There we go. So let's go back to the starting page for this. Move it up. Okay, great. What you're going to do on this page is you just need to get to the list of all the genes. I'm sure there's another smarter way to do this. I've been just typing the letter A. It seems to be in most of them. Hitting return, waiting a second, and then it'll bring us to the list of all of the conserved orthologous genes. Here we are at the list. Okay, so each of these little groups, COG 0001 on through, I think there's like 3,000 or 4,000 of them. These are conserved orthologous genes that you find in lots of different organisms. If you want to see the categories, they are here. This tells you what they do. So if it's an N, it's going to be involved in cell motility. If it's an H, wait, let's go up here and see. H, it's going to be involved in, there we go, coenzyme transport and metabolism. Marvelous. If you care, that's what they're involved in. Let's see. I'm going to go down here to the cadherin family, which is going to be 1456, and it should be already hit. There we go. So you're going to pick one at random. I'm picking this one. Don't pick this one. Okay, take you to this. And first you want to check. You want to look and see, all right, in the archaea and in the bacteria, do I have representation of this gene? Otherwise, it's not going to be good for this assignment. We want something that has really broad representation. But here I see, these are in the archaea, and awesome. There's a bunch of them. Most of those are eukaryotes. Go down here. Are there any? Yes, Chloroflexi. All right. They have some, and you get a bunch in Clostridia. Ew, yeah. That's like C. diff. And Delta Proteobacteria. Okay, so they're all over the place in bacteria and in archaea. That's great. So now we know they're good, and so now let's go and click on the COG symbol. Yours will be different, but it'll look like this, and it'll be in this place. Give it a second. There we go. We've loaded up. Okay. This little list, all these little links at the side, are a particular version of this gene. So these are all built on the same ancestral protein fold. They share a common structure and a common ancestor, but they've evolved a couple of different functions now. Anyway, one at a time, you're going to click on these things. And so let's click on this one, and let's click on one from bacteria so you can see how to handle those. Here, let's try that one. All right, so let's look. Okay, so for this one, you got two choices. One, you can do a little bit of mousing over. So if you mouse over this, it will eventually show you that you can blast the protein, and there you go. That's the number we want. WP, whatever, 011, and so you copy that. So that's one way to do it. For some genes, you can go to the transcript table, and there'll be something there. For the bacterial genes, that does not seem to work, unfortunately. But for the eukaryotic genes, that seems to work very nicely. So here, I think this is a eukaryotic version. And yeah, so there we can just copy the protein. The protein is probably going to start with either NP or XP or maybe even WP. And that'll just tell you the P, second letter, is what tells you that this is a protein sequence. All right. So you're going to copy just those names. And you can even leave off the 0.1, 0.2, 0.3, whatever at the end, because that is not important. So you're going to make a little list of those, and yeah. All right. So basically, and then you're going to go back and you're going to do that again. You're going to go back to this page. We already did the first one. We'll go to the second one, third one, fourth one, until we get 10 of these things. And we would like to have them from a mixture of eukaryotes and bacteria. How do you tell? Well, you can Google it. But you can also look, like this one says Xenopus laevis. Yeah, all right, but it's an African clawed frog. Then you can think back and say, oh yeah, frogs, they're eukaryotes. That sounds good. And a lot of bacteria have names that end in obacter or ococcus. Those are a good indication. And archaea are chaotic, so whatever, deal with those if they come up, as long as we have a mixture though of at least 2 of the 3 domains of life. I think that's going to be fine. There aren't a ton of archaeal genes out there. Anyway, back to the thing. Like, okay, done with that. So type any letter. I typed E, whatever. Here, I typed A that time, so there we go. Hit return, click the links, choose a cog, blah, 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 choose a symbol, get your transcript table. There you go. Here's your transcript table. And so let's do the report. So the report wants a sentence about its function and what its name is. So that's its name, cog 1456, CDHE, that's the thing we clicked on, which stands for cadherin type E, which is the human equivalent of this. It's related to these iron sulfur cluster proteins, which is what that page said, but you can also Google it. And if you Google cadherin E, you find out that it's used by eukaryotic cells to maintain cell-cell interactions. It's basically one of the things that lets us be multicellular. So it's pretty conserved in multicellular things, but you also find it being used for a different purpose down in prokaryotes, which is kind of neat. So there's my list, a mix of NPs, XPs, and WPs. Good job all the way around. Here is the screenshot. So I ran these through BLAST. So let's try that. Let's see. If we were to go to BLAST, there we are. And we need to do protein BLAST because we've got protein sequences. And then let's just grab all of these. Got them. All right, and let's put them in. So by default, BLAST is going to come up and it's going to look like this with only one box here. You're going to click this thing to align 2 or more because that's what we want to do. And that's fine. You're going to paste the first one in the first thing. So that's great. That one can live there. You're going to paste all the rest in the next thing and you're going to hit BLAST. You're not going to mess with anything else. That's going to be good enough. You're going to wait a second. There you go. We got our BLAST back. And boom, screenshot number 1. This screen right here. I want to see those stats so we can see if these are good or not. And 2 times 10 to the minus 139 is that's a pretty low chance of error, so I like it. Then we're going to go over here on the alignment page. It's going to default to say alignment view pairwise. But what I want is flat, query anchored, and it's probably better with letters for identities, but you can do dots if that makes you happy. And that just shows all of them together. Boom, just like that. Okay, looking good. And then I can't screenshot all of it because it's too big. It's bigger than a screen. So I'll just screenshot the top 1 or 2 rows and that'll be good enough. And so paste those in the report. There's my stat page. There's my alignment page. And then the last thing is a sort of a 1-sentence analysis. So we've got really good stats on 3 of them, but the other sequences align so badly that BLAST excluded them from the stats page. And you may find the same thing. They may not show up, in which case it just means that they are not close enough for BLAST to be certain that they are actually homologous. There are some homologs in the world that are not readily apparent at first. It takes other techniques, and BLAST is fast, but BLAST is not always the best. All right, so none of those appear on the alignment page. Let's take that out. More than one part of each sequence aligns. What I mean by that is that this one that ends in 439, you see it's on there twice. That's two different parts. You can see from 293 to 343 and from 513 to 563. They both aligned with my query sequence, which is the one that I was trying to align them to, which is up here. And so what that would indicate is that this protein may have duplicated domains, like it has two parts in it that actually align with statistical confidence to the same thing. So there you go. It's a double cadherin. Who knew? Yeah, I guess people that work on that sort of thing probably already did. Okay, there you go. That was the very short rundown on how you do this. Try it yourself. Try it with my example if you want. And then pick one of your own and try it. And as long as yours is different from all your friends, everything's cool. Turn it in via Canvas and you're good. Thanks.